We have seen tiny plants that grow in moist conditions as well as big trees. But plants bearing flowers are not included in any of the groups. In this video, we will be learning about the plants which produce flowers, that is angiosperms. The word angiosperm originated from two Greek words, angion which means vessel and sperma which means seed. The seeds are enclosed in a vessel, that is fruit wall. Even though the gymnosperms and angiosperms are vascular structures, they differ in the seed structure and the arrangement. Let us check one by one. Angiosperms are seed plants in which the pollen grain and the ovules are developed in specialized structures called flowers. And the seed is produced within the fruit wall. Angiosperms occur in a wide range of habitats and in different sizes. The smallest angiosperm is Wolffia, which is a small aquatic structure. The tallest angiosperm is Eucalyptus. Angiosperms have various commercial and economic values. First, let's study about their seeds. In gymnosperms, a seed is produced by non-flowering plants and the seeds are unenclosed or naked. In angiosperms, the seed is enclosed within the ovary. Based on the number of cotyledons present inside the seeds, the plants are divided into two classes, that is monocotyledons and dicotyledons. So, what is the purpose of cotyledons in plants? Cotyledons store reserve food materials. So, now let us check the various features of monocots and dicots. Monocots have a single cotyledonous seed, leaves which shows parallel venation, and the flowers are trimerous flowers having 3 members in each whorl. They exhibit fibrous root system. Grains, lilies, daffodils, sugarcane and grass are the examples of plants that are monocots. On the other hand, dicots are the plants that have seeds that have 2 cotyledons and their leaves show branched or reticulate venation, and they produce tetramerous or pentamerous flowers, that is having 4 or 5 members in each floral whorls. They exhibit a taproot system. Legumes such as pea, beans, peanuts, mint, lettuce, tomato and oak tree are few examples of dicots. Let us now focus on the reproduction of angiosperms. The flower is a reproductive organ of angiosperms. The male sex organ is the stamen. So now let's focus on the structure of a typical stamen. The stamen consists of a slender filament and an anther at its tip. And within the anther, the pollen mother cell, that is a PMC, divides by meiosis to produce microspores which matures into the pollen grains. So we can infer that pollen grains are the male gametes. The female sex organ in a flower is a pistil or a carpel, which consists of an ovary enclosing one to many ovules. So, a pistil consists of an ovary, style and a stigma. You can see the embryo sacs within the ovules, right? And the embryo sacs are highly reduced female gametophytes. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divides to form the embryo sac. The embryo sac development is preceded by meiosis. So, all the cells of the embryo sacs are haploid. Each embryo sac consists of 3-celled egg apparatus, 3 antipodal cells and 2 polar nuclei. The egg apparatus consists of 1 cell, that is one egg cell and two synergids. The two polar nuclei eventually fuses to produce a diploid secondary nucleus. Recall the concept of pollination. The pollen grains are dispersed from anther and they are carried to the stigma of the pistil. So we can define pollination as the transfer of pollen grains from the anther to the stigma. So transferring pollen grains are carried by pollinating agents, right? They are like wind, water, insects, birds and bats. Once a pollen grain of the same kind reaches the stigma, they germinate to produce a pollen tube and the two male gametes are formed in it. The pollen tube reaches the ovule through the tissues of the stigma and the style. The pollen tube enters the embryo sac and the two male gametes are discharged. Thus, one male gamete fuses with the egg to form the zygote. This process is termed syngamy. The two polar nuclei, that is N plus N, fuses to form a diploid secondary nucleus, that is 2N. The other male gamete fuses with the diploid secondary nucleus to produce a triploid, that is 3N, that is primary endosperm nucleus, PEN, called triple fusion. It is through the process of triple fusion. The two triple fusions, that is syngamy and the triple fusion in the same embryo sac is termed double fertilization. Now, let us check the post-fertilization events one by one. The zygote develops into an embryo with one or two cotyledons. The primary endosperm nucleus or the PEN develops into the endosperm. The endosperm provides nourishment to the developing embryo. Synergids and antipodals degenerate. The ovule develops into a seed while the ovary develops into a fruit. Does the haploid and diploid stages are found in all plant species? Have you checked it? Let us check the same in the next video. So let us summarize what we have learned.
Manorama Horizon. Learning made simple.